Hello and welcome, students! As you join us in the lab today, we're about to delve into a fascinating hands-on experiment. So far, we've explored how various factors such as temperature, water, and the availability of oxygen play crucial roles in the rate of decay of biological material. You've learned that gardeners and farmers harness these variables to optimize the conditions for rapid decay of waste biological material, creating compost that acts as a natural fertilizer to nourish garden plants and crops. Today, we're going to apply this theoretical knowledge in a practical setting by investigating how temperature affects the rate of decay of fresh milk. We'll measure the changes in pH over time as an indicator of decay. This specific experiment will help solidify your understanding of how temperature can influence the decomposition process. By the end of the experiment, you'll not only have first-hand experience observing the rate of decay, but also see how manipulating environmental factors can impact decomposition, similar to how gardeners and farmers create optimal conditions for composting. So, let's get started and dive into this practical exploration of decay. Before delving into the specifics of this practical, it's crucial to understand why investigating the effect of temperature on the rate of decay of fresh milk by measuring pH change is valuable. Here are several key motivations. Understanding the rate of milk decay has direct applications in food safety and storage. Milk is a common household item, and knowledge of how temperature affects its spoilage can help you make informed choices about how to store it safely. This practical allows you to connect classroom concepts with real-life situations, enhancing your understanding of why certain foods need refrigeration and how temperature management extends their shelf life. Secondly, this experiment provides insight into microbial activity and enzyme function. This experiment simulates microbial activity and enzymatic processes responsible for decay. By measuring pH changes, you observe how enzymatic processes in milk produce acids as metabolic byproducts when exposed to varying temperatures. This understanding is fundamental in fields such as microbiology, biochemistry, and environmental science, providing a foundation for more advanced topics like enzyme kinetics and microbial ecology. Finally, conducting this experiment hones your ability to design and execute a controlled scientific investigation. You'll learn how to manage variables, systematically collect data, and interpret results. Skills like accurate pH measurement, data recording, and graphical analysis are essential not only in academic settings, but also in many scientific careers. This practical, therefore, offers you a hands-on opportunity to practice and refine these crucial abilities. In summary, this practical is not only about observing milk spoilage, but also about connecting theoretical knowledge to practical applications, understanding microbial and enzymatic activities, developing essential scientific skills, and fostering critical thinking. Let's get started. Let's walk through the steps together. First, let's prepare our lipase preparation. Begin by measuring 5 centimeters cubed of lipase solution and pour it into a test tube labeled L. Next, measure 5 centimeters cubed of fresh milk and place it in a separate test tube labeled M. After this, take some phenolphthalein indicator and carefully add 5 drops to the milk test tube. Next, carefully measure 7 centimeters cubed of sodium carbonate solution and transfer it into the milk test tube. This will make the solution alkaline and turn it pink. Now, we set up a water bath and adjust it to a steady 30 degrees Celsius. Place both test tubes, the one with lipase and the one with milk, in the water bath. Allow the test tubes to sit in the water bath until they reach the desired temperature. Use a thermometer to ensure the milk reaches 30 degrees Celsius. Once the temperature is stable, use a calibrated dropping pipette to add 1 centimeter cubed of the lipase solution to the milk test tube. Immediately start the stopwatch and stir the mixture with a glass rod to ensure the enzyme is uniformly distributed in the milk. Carefully observe the solution for changes. The pink color will gradually disappear as the solution becomes acidic. Stop the stopwatch as soon as the pink color completely disappears and record how long this took. Repeat the entire process for different temperatures, such as 20 degrees Celsius and 40 degrees Celsius, to see how temperature affects the rate of decay. Conduct the 3 trials for each temperature setting to ensure consistency. Average the times recorded for each temperature to enhance reliability. Let's now turn to our analysis. The central biological reaction investigated in this practical is the breakdown of milk fats and proteins by the enzyme lipase, resulting in the production of fatty acids and glycerol. This reaction decreases the pH of the milk, making it more acidic. By measuring these changes in pH at various temperatures, we can study how temperature affects the rate of decay. Now, let's take a look at the key steps and their reasoning. Firstly, we carefully prepare the solutions. Adding 5 centimeters cubed of lipase to a test tube ensures that we have a controlled amount of enzyme ready for the reaction. The fresh milk in the other test tube is our substrate. Adding phenolphthalein and sodium carbonate solution makes the milk solution alkaline and pink, which prepares it for the experiment by allowing us to visually track the pH change when lipase acts on it. Secondly, we carefully set up the temperature. Placing the test tubes in a water bath set to a specified temperature ensures that our reaction proceeds under controlled and accurate temperature conditions. Using a thermometer to monitor the temperature helps us ensure that the milk has reached the desired experimental temperature. Thirdly, we started the reaction by adding lipase to the milk. Adding lipase to the milk at a specific temperature and starting the stopwatch allows us to time how long it takes for the enzymatic reaction to occur, causing a visible pH change. Next, we monitored the physical reaction. Continually monitoring and timing when the pink color disappears enables us to measure the precise duration of the reaction for each temperature setting. This data is crucial to determine how temperature affects the rate of decay. Finally, we repeated the experiment. Performing the experiment at multiple temperatures helps us measure the effect of different temperatures on the rate of decay of milk. Conducting 3 trials at each temperature ensures the accuracy and reliability of our results. Repetition helps minimize the influence of anomalies and improves the validity of the data. Now, we need to model and analyze our results. For each temperature that we conducted the experiment at, record the time taken for the pink phenolphthalein to turn colorless, indicating a pH change. Record these times for each of the 3 repeats. From these times, calculate the average time taken for the solution to change color. Next, we need to calculate the rate of decay using the formula. Rate of decay equals 1000 divided by average time. This gives the rate of decay in units per second. For example, if the average time at 30 degrees Celsius was 120 seconds, the rate of decay would be 1000 divided by 120, which equals 8.33 seconds to the power of -1. From this data, create a graph with the temperature on the x-axis and the rate of decay on the y-axis. This visual representation helps identify trends and relationships between temperature and the rate of decay. By examining the graph, we can determine how temperature impacts the rate of lipase activity. Finally, we need to discuss our results. In the graph, we could see how, as temperature increases, the molecules move faster, resulting in more frequent collisions and hence increased reaction rates up to an optimal temperature. Beyond this optimal temperature, enzymes can denature, leading to a decrease in the reaction rate. We should also consider any anomalies in our data. Factors such as precise measurement of solutions, accurate temperature maintenance, or consistent stirring could influence results. Discuss any potential sources of error and how they might be mitigated in future experiments. In today's lesson, we conducted a practical experiment to investigate how temperature affects the rate of decay in fresh milk by measuring the change in pH in response to breakdown by the enzyme lipase. The topics we covered included the process of decomposition and its role in the ecosystem, the influence of temperature on the rate of microbial activity and decay, and how changes in pH can indicate the progression of decay. Our key takeaways for future practicals are to ensure precise control of variables like temperature and time intervals for reliable results, use accurate equipment to measure pH changes consistently, and recognize how environmental factors, such as temperature, affect the rate of decay and the importance of monitoring these changes. This practical not only reinforced our understanding of decomposition and microbial activity, but also highlighted the importance of controlling experimental variables and accurately measuring pH, to study biological processes effectively.